Good morning, dear friends. This is the second lecture about the vectors, DNA vectors that I was talking about. In the first lecture, we discussed about the plasmid vectors. Here in this lecture, we will talk about the other vectors like these Phage vectors and hybrid vectors like Cosmids and Phagemids, and a couple of high capacity cloning vectors also. So let's begin with this Phage vector first. As you can see that these Phage vectors have the capacity. These Phage vectors are designed to clone inserts more than 6 kb, 7 kb, 8 kb, up to maybe 25 kb DNA inserts could be cloned. For that purpose, these Phage vectors were. Phage vectors have also this origin of replication. They are self-replicative. So this is the Lambda DNA vector. It is a double-stranded linear molecule, as you can see, about 48.5 kb kilobases long, approximately 50 kb you can say, and it has it can broadly be, as far as the vector is concerned, it can broadly be divided into three regions: the left arm, which is about 20 kilobases, the right arm, which is about 9 kilobases, and in between them there is a region, the middle portion, which we call the stuffer DNA, because you can replace this stuffer DNA with your own stuff of DNA, the insert, and its size is about 14 kb. So you can incorporate your gene of interest, which may range in size from 9 kb to 23 kb, like that. So this portion of the DNA can be replaced, and that's why the name is the replacement vector. This portion is, if you replace this portion of the DNA with your own desired gene, then what will happen? Some genes present within this region will be lost. But there is no problem, because the genes that are present in this region are basically required for lysogeny, for recombination of the lambda phage within the host chromosome. So that is not desirable as far as the vector is concerned. For a successful vector, it has to be a lytic. It has to go for lysogenic cycle, lytic cycle, so that you can produce plaques, plaque forming units. From there you can screen the plaques by plaque hybridization, or you can directly pick up the plaque in which you have your desired gene of interest you have selected. Anyway, so you can replace this stuffer DNA with your gene of interest. The upper portion, the upstream portion, has head and tail and other parts of the structural genes that are present in this genome. The right arm portion has the genes for the lytic cycle and also for the multiplication or replication of the lambda DNA. And these ends are very important. They have, you can see here, the cos sites on both ends. These cos sites are responsible for packaging of the phage genome inside the capsid. When the replication of the phages is going on inside the host cell, in lytic cycle, what happens? The phage genome ultimately gets packaged into the whole capsid, and for that you require these cos sites. And interestingly, this half cos site and half cos site both are split in this manner, so they are not going to be packaged. If the genome is present in this manner, it is not going to be packaged. So as soon as the the DNA enters into the host cell, it circularizes with the help of these single-stranded region. These are basically, on the two sides there are 12 nucleotide long overhangs, and they are cohesive ends. They are sticky. They are complementary to each other. So when the DNA folds back and it hybridizes with this 12 nucleotide overhang on the other side, the nicks that are present on the other strands, they get sealed up, and the molecule becomes circular. This circular molecule even cannot be packaged, even when the cos sites have been united and the full cos site is present in the circular molecule. But this will not be packaged, because for packaging to occur, it requires 1.5 cos site. But in a circular molecule, there is only 1 cos site. So why this circular molecule is formed at the beginning? Just to increase the number of the copies of this DNA. It undergoes a theta mode of replication at the beginning. Then it undergoes transcription and translation, synthesizing the capsid and other parts of the phage. And when the time of packaging comes, then the circular molecule, it undergoes a rolling circle mode of replication. So when it goes for rolling circle mode of replication, it replicates several genome length DNA. Those are ligated end to end, forming what you call multiple genome length DNA. They are being continuous. They are associated end to end because of these cos sites, and that structure we call concatemer. So chain of genomes. This concatemeric DNA, it will have 1.5 cos sites. You can see at the termini there is half cos site, and within the genome there are full cos sites. So when the first portion will be packaged by a mechanism that is called headful mechanism of packaging. We will talk about packaging in some other lecture. That is very interesting. Not only in lambda phage, there are other phages in which packaging is very, very interesting phenomena in molecular biology. So here, when the first segment will be packaged, then it will leave half cos site over at the end again, and there is a full cos site within the genome. Then in the next, when the next genome is there, at that juncture there is a full cos site. So again, this will also get packaged in the other capsid. So in this way, the entire all the capsids will get packaged with the genome length size DNA. So this cos site is very, very crucial for these lambda phage vectors. So this is a replacement vector. It is called replacement vector because you are going to replace this middle part with your desired gene of interest. For doing that, you need to have some restriction sites over here at this juncture that are present. So when you cleave with those sites, you generate 3 segments: this left arm, this middle part, and this right arm. So these are the mixture of DNA. You need to separate them by agarose gel electrophoresis, and you need to purify them. You need to elute these portions, the left and the right arm. You don't require this middle part. So you purify the left and the right arm, and you add your gene of interest with it, and you allow it to ligate. So what will happen? There are 3 results that might happen. The left arm and the right arm might get united, that might get ligated, forming what is we call the non-recombinant DNA, and they will also form a concatemer, but the cos to cos distance will be only 29 kb. The interesting feature of these phages, lambda phages, are 2 mainly. One is this: you can replace this middle portion. That is one thing which gives you facility to incorporate your gene of interest over there. The other thing is this: the phage genome, this genome will only be packaged if the total size, if the cos to cos distance is about 40 to 50 kb. It may go up down to 38 kb, or it may be 53, 54 kb maximum. But in general, 40 to 50 kb, if the distance between 2 cos sites is within that range, then only the genome is going to be packaged, otherwise not. So vector to vector, left arm and right arm, if they become ligated, what is the size? 29 kb. So these 29 kb, 29 kb, 29 kb segments are not going to be packaged. So you are getting rid of the non-recombinant clones automatically. They are not going to be packaged in the capsid, so they are not going to produce any plaques. They are not going to lyse the host cell. So I will tell you how plaques are formed. So they are not going to form plaques, but the other result is what? These inserts they have also the similar ends, so they may also get ligated one after another, forming a very long molecule. So but they don't have the cos site, they are not going to be packaged, and they don't have the other genes also. For there is no question of them being, even if they are not going to be packaged first of all, and they don't have the other genes for lysis and other head, tail formation, etc. The third one, which is the desired one, is this: that the left arm and the right arm, they will contain an insert in between them. So this is the right thing that you require, the recombinant DNA, and they will again form a concatemer. So the cos to cos distance will automatically be, if the insert size is about 9 to 23 range has been given in the textbooks and literature that it may range from 9 to 23 kb, but I am considering it a little bit maybe 12 kb to 20 kb say. So these size inserts will easily get packaged if this insert size is that, then what is the cost to cost distance? 20 kb plus, say it is 12 kb, 20 kb plus 12 kb is 32 kb, and then 9 kb is 41 kb. So cost to cost distance, if the insert size is 12 kb, is 41 kb. This DNA is going to be packaged in the capsids. So these capsids will finally lyse the host cell and will form plaques in the petri dishes. When you will plate these infected bacteria with this recombinant DNA, those cells will get lysed, and the new phages will infect the nearby cells, and they will again lyse those cells. Like this, if you plate them on a lawn of a bacterium, then what will happen? They will form small clear areas. Those are called plaques. And if you delay a little bit, if you leave that plate like that, and if you allow to grow them further, then what will happen? Each of the plaque will grow. They will continue lysing the surrounding cells, and the plaques will merge together, and the whole plate will become empty. They will eat up the entire bacterium. So that is not desirable for cloning purpose. We have to go in the morning. You do this in the evening, and you go early in the morning to lift those plates so that the distance between plaque to plaque is suitable for your further screening purpose. You want to lift the plaques, or if you want to go for library screening. For that purpose, you need to have a distinct plaques in your plate. So you don't allow the plaques to increase in size further, so that they merge up together. Okay, so this is how these replacement vectors are very suitable. The recombinant selection is automatic, because whatever plaques you are getting is because of the recombinant clones. The non-recombinant are not going to be packaged. So this particular lambda replacement vector is used for genomic DNA library preparation, and there are different examples like the lambda-2, the Sharon phages, CHARON, Sharon 3, Sharon 4. There are EMBL3, EMBL4. These vectors are replacement phage vectors. The other version is the insertion or integration vector. This is an alternative form of the same. In this case, what scientists did is that they have removed this tougher DNA already, and they have made a ready-made vector so that you can directly go for your restriction digestion and cloning without going for the gel electrophoresis in the middle. So that is in fact limiting your time. That is delaying the process. So they thought to remove this portion and to ligate the left arm and the right arm so that they can directly be used. But what happened after making this sort of vectors was that the insert size that can be cloned within these vectors is little less than what you can clone in the replacement vector. These vectors can hold 6 to 9 kb size inserts. And here there are two examples, lambda GT10, lambda GT11. Both are of a different concepts as far as the screening of the recombinant is concerned. This contains the C1 plus gene, whereas this contains the LAGZ gene. And these genes are, there should be an arrow over here. These genes are interrupted with these multiple cloning sites. So when you clone your gene of interest within them, you disrupt either the C1 gene or the LAGZ gene. Both of these genes are required for the purpose of recombinant selection. How? C1 gene is basically, it is a lysis repressor. Lysis repressor means what? If C1 gene is intact, it will not allow the phage to go for lysis. It will allow the phage to go for lysogeny. So the plaques generated by the lysogeny will be hazy. You will be able to distinguish those plaques from the lytic plaques. They are clear plaque forming units. So if your C1 gene has been disrupted by the gene of interest, then the C1 gene will not produce the C1 protein, and lysis inhibition will not occur, and the phage will go for lysis. So the clear plaques are the indicative of the recombinant clones. And you already know about the LAGZ gene, that it is required for blue-white screening. So you have to put XGAL and IPTG in the media, so that your recombinant clones will be white, your non-recombinant clones will be blue in color. So you can distinguish them like that. You can lift your recombinant clones from there. Okay. So these two vectors, lambda GT10, lambda GT11, they are insertion vectors, and they are used mainly for cDNA libraries, for making cDNA libraries. cDNA are only the expressed region of the genome. They don't contain the non-genic region. They neither contain the intron part even. They are made from the mRNA. So the processed mRNA, I mean the final mature mRNA that does not contain any intron within them, they are spliced and they are almost already processed. They have been brought to the cytosol for protein synthesis. So from there you are making this DNA. Those are called cDNA. And when you are cloning them in this library, in these vectors, they form the cDNA library. Okay. So this is about these Phage vectors. Now let us go for Cosmid vectors. Cosmid vectors are one type of hybrid vectors. Here you can see the cos and mates. Cos is from the cos site of the Phage and mates is from plasmid. So the rest of the portion is plasmid backbone, particularly the P called E1, but the origin of replication is of F1 origin of replication. The cos site has been provided here so that this DNA can go for packaging also, if the cos to cos distance is about 50 kb or so. So, but the size of this plasmid is about 5 kb, Cosmid DNA. So when you digest at the multiple cloning site, it becomes linearized like this. And when you add the inserts, which range from 40 to 45 kb size, so you are able to clone a little longer inserts in these vectors. So what happens here, again, there are three situations that the vector to vector, they get ligate together, but this cos to cos distance will only be 5 kilobases, so it will not be packaged in the Phage capsids. The inserts, if they get ligated, they don't have the cos site, so they will automatically reject it. The other one, the recombinant concatenameric forms like this. This is the vector DNA. This is another vector DNA, and you have these inserts have been ligated within them, in between them. So from here to here, the distance becomes this cos site. This is the cos site, and this is the cos site. These blue, light blue regions are the cos sites. So from cos to cos, the distance is about 40 to 50 kb long, 45 to 50 kb long. So this DNA is going to be packaged inside the capsids, and they are going to produce the plaques finally. This is the lawn of the bacterium in the petri dish, and these are the clear zones. These clear zones are the plaque, we call it plaque forming units. Okay, so phages don't produce colonies. They will lyse the bacterial cells that are present. So first you generate a lawn of a bacterium, and some of the cells, if they are infected, they will continuously eat up their surrounding cells, and gradually they will form a clear zones like this. And this will continue, as I was saying, that if this circle increases in size, this circle increases in size, this circle, all of them, these plaque forming units, if they increase in size, they will merge together, and the entire plate will be empty after some time if you leave it, if you allow it for growing further. So that is undesirable, of course, for the purpose of screening. So this is, Cosmic vectors are also used for genomic DNA library preparations. Now coming to the Phagemids, these are also one type of hybrid vectors. Here also Phage and mid, I mean the plasmid, two backbones have been mixed up. How, let us see, and why they have been made. They are not made for cloning larger size inserts. Rather, they have been designed for making single-stranded DNA molecules, or for doing site-directed mutagenesis, SDM. So let us see, there are two versions here. The one is the pBSSK+, plasmid BlueScript 2 SK+. The SK+ name comes from here, the multiple cloning sites that terminate with this SAC1 and KPN1. So you take S and K from here. The other version will be KS+, in which this portion will be in inverted position. And this is a transcription vector also, because this MCS contains two promoters, T7 and T3 viral promoters, Phage promoters. So the insert that you are going to clone here, you can easily produce transcripts of that insert by using T7 RNA polymerase in vitro reaction. I mean, you have to purify the DNA, and then you have to cleave this DNA by some restriction in the nucleus. Say if you cleave it by KPN1, your insert is present here somewhere, so it will become linearized. And if you are using this T7 RNA pol, you are going to transcribe your gene of interest over here. This is called runoff transcription. This is required for people who are working with RNA. So this vector is very useful as far as the transcription is concerned, and also you can clone your gene here just for the purpose of storage, that you are keeping your gene in a particular host cell for some other purpose maybe. And when desired, you can go for producing single-stranded DNA from this, because this contains a Phage origin of replication, which is this one, F1 origin of replication. The F1 phages are single-stranded DNA phages. So if you co-infect these E. coli hosts that contain the recombinant pBSSK plus genome, I mean the DNA vector, recombinant vector, if you co-infect with this F1 phages, what will happen? This particular DNA, which was double-stranded, will now produce single-stranded DNA molecule. That phage will act as a helper phage. It will, because this particular DNA does not have the other genes of the phage, only the origin of replication, so it is not able to replicate a single-stranded DNA molecule. When it gets the other DNA, other genes required for its single-strand DNA synthesis and phage capsid formation and other processes, then it undergoes single-strand DNA synthesis, and that is required for sequencing purpose, particularly the Sanger's dideoxy chain termination sequencing method that requires single-stranded DNA molecules. This is M13 single-stranded DNA phage vector, and this contains, both these vectors contain the LAG-Z gene, so they can go for blue-white screening, of course. And here there is a universal primer sequence. This is very interesting. This particular vector is required for producing single-stranded DNA molecule for sequencing on one hand, and the other most important thing is this is used for site-directed mutagenesis. You can use this universal primer, you can attach, you have cloned your gene of interest in this multiple cloning site first of all. So if you want to make some alteration, some mutation in your gene, you want to change a single nucleotide base pair, maybe from AT to GC or from GC to AT, whatever, or you want, maybe you want to make several nuclei, quite a few, means an oligonucleotide, maybe 7, 8, 9, 10 nucleotide long sequence. You want to change that, in fact, or you want to change only one base pair, you can go for this. The single nucleotide point mutation that we do by this method is called site-directed mutagenesis, because here the mutagenesis is very, very directed at a particular site. It is not random as it is used in case of generating bacterial mutations by nitrosoguanidine and other methods, UV method or other methods, by using mutagens. Those are random, and the screening process is difficult in that case. Here you have a specific mutation you are introducing at certain nucleotide. Okay, sorry. So what you can do, you can make a single-stranded primer. You can attach that primer with this one, okay, and that primer will have that particular change. Say if there is A in the gene and you want your new particular genome that is going to be formed, the particular recombinant DNA that is going to be formed from this to be altered, in place of A you want AT, you want to convert it into GC. So what you take in the primer, just opposite to A, you take G instead of T. The other nucleotides will be complementary to the gene, because you want to change only that position. So now your primer, when it will form a new DNA strand by primer extension, the complete circle. And again, when these two DNA strands will replicate to form yet another double-stranded DNA molecule, then what will happen? The inner DNA will result into a correct double-stranded DNA, because it will replicate and it will bring T against A. But the other strand, which is, you can imagine that is outer strand, but actually the double-stranded DNA are in a helix form, but you cannot say which one is outer, which one is inner. Here in the diagram, just for our understanding purpose, the outer strand that contained the G, it will bring a C in its opposite position in the complementary strand. So these two DNA molecules will move into two different host cells, and after that you need to screen them which particular contains the GC at that position where you desired. So this is your point mutation that you have introduced. Now you can study with that particular clone whatever you want to study. You might want to see the effect of the change that you have made in terms of the activity of the enzyme that is being expressed from that particular gene, or it may be some other purpose. If it is you are studying with some RNA, so you may go for the structural changes that it may bring in the RNA molecule. Anyway, so site-directed mutagenesis can be done by these vectors. So now coming to the high-capacity cloning vectors, we will talk about two such vectors, YAC and BAC. These YAC vectors is artificial chromosome. It was developed in 1983 for the purpose of mainly genome research, genomic research. It began in Human Genome Project particularly. The first genome probably was sequenced was Haemophilus influenzae, I believe. And then Human Genome Project was also going on. It started in 1989 and it was finished in 2003. It took such a long time. But nowadays you can do it in a week. You can annotate even within a week. But it took a long time because the method that was used was the Sanger dideoxy method, and nowadays you use the next generation sequencing, a totally different method of sequencing. So though there were very high throughput machines that were used, it took such a long time. At the beginning, these YAG vectors were used. Later on they shifted to BAC vector. In fact, there were two groups. One was the federal group, those were using these YAG vectors, and the private run group led by the Craig Venter, who was also a part of the federal group till 1995, but because of this particular issue that he was disagreeing with this, going with this YAG vector strategy, he wanted to use the BAC vectors. He separated himself from there and he joined a private company called Cellular Genomics. And both these groups completed sequencing. They published the draft sequence in 2003. One group published in the Science journal, the other group published in the Nature journal. And they had more or less they had discovered about 31,000 genes at that time or so. Nowadays we know that there are about 21 to 22,000 genes. There is a little bit of dispute about some genes. We will talk about these things later on. So YAG vectors are very suitable for genomic research, but they have also some problems. They can clone very big sized DNA molecules, about 1.5 megabase pair length DNA molecules, which are very big. And so when this particular vector is relatively small, 11.4 kb only, but it can hold DNA of that size, so the final size becomes a chromosomal size DNA. So these vectors have interesting feature that they have telomeres. There are two telomeres and there is a centromere, because this is a eukaryotic vector. It has to reside in a yeast cell, so that's why for proper separation to occur it requires a centromere. Telomeres are responsible for the maintenance of the chromosome, and the function of the telomere. There are three important functions. Here I will not discuss those. So in this BAC vector, when you go for cloning, you have to digest it in this BAM site so that this become linearized. like a linear chromosome of an eukaryotic system, and at the same time you can digest with either EcoR1 or SNAB1 in this SUP4 region, which is a marker gene for selection, recombinant selection. URA3 is also a one marker gene. So your gene of interest is cloned here in the SUP4 region, and you can this is the result of the vector when you digest it with BAMH1 and SNAB1. And these are the inserts that also being digested with SNAB1 or EcoR1. If you use EcoR1 here, you have to use EcoR1. And then when you ligate, this is the recombinant that results with the two telomeric regions here and the centromere somewhere, which is a centromere. Centromere is this color. Rs, this portion is the centromeric region, you can say. This Rs is the automatic replication sequence. This is required for, this is one origin of replication that is needed. AMP gene is also there. Origin of bacterial origin of replication is also there, because this can also be maintained in bacterial cells for the purpose of its storage or maintenance. So this is how these YAG vectors are used for genomic DNA research. This YAG vector, the structure of the YAG vectors is like this, and the problem with the YAG vector is that this long side insert that they clone. On one hand it is useful, but on the other hand, what happens, because of the long size of the insert, there are several repeated regions in the genome, in the particular DNA that you have inserted, and those repeated regions are complementary. Repeated means they are complementary, so they may hybridize together and they may generate different types of gene rearrangement. You may get deleted portions of the gene, you may get inverted portion of those genes, or repeated portions of the same genes. So this sort of gene rearrangement is undesirable when you are doing sequencing, genome sequencing. You will misinterpret your genome. So that's why YAG vector later on become unpopular as far as this genomic research is concerned, and BAC vector took its place. It was developed a little late in 1990s, and this vector can hold DNA of about 100 to 300 kilobases size, which is quite good for as far as the chromosome walking is concerned. So when you are doing genomic research, you need to have chromosome walking so that you can quickly run a whole length chromosome first. You have to break that chromosome into longer fragments so that you can map that physically, that which fragment is present after which, and then you need to have some smaller libraries, smaller DNA libraries for the purpose of sequencing, for knowing for completing those long segments, because you cannot sequence more than 1 kilobase, or hardly you can go for up to 1.2, 1.3. The error starts after 1 kb. So considering that 1 kb as the standard size for sequencing, so you need to have smaller size DNA. Several different kinds of libraries are being made for genomic research. Genomic research is another thing that is also very interesting, but these vectors are basically made for those purposes. This vector contains the PAR and PAR B gene. These are responsible for partitioning of the chromosome when the bacterial cells are dividing, since they don't have any centromeric region. So the function of this PAR B is equivalent to those centromeres, you can say. So these are not telomeres, these are the sp6 and T7 promoters. So insert that you are going to clone here in the multiple cloning site, you can transcribe those inserts. The end region of the inserts can be transcribed, of course. So it is a kanamycin resistant gene for selection purpose, and there are origin of replication, and there are some other gene like REP-E, etc. So this is how these BAC vectors look like. Okay, so this was all about the different types of vectors that we were talking about, and one more type, that is TI plasmid based vectors, those which are required for plant genetic research, genetic engineering. I did not include that here because I will make a separate vector with that, and along with the plant genetic engineering part, how that is being used for that purpose. That is a little complicated vector. There are two types of them, so we will have a separate lecture on that. So here I'm going to end this lecture. I think as far as your undergraduate syllabus is concerned, this is more than enough. and if I go further, it will become uninteresting. You can consult different books for this purpose. You can consult articles, you can see other videos also of experts. So thank you very much. I think you like this video, and if there is any issue, any question, any controversy, whatever, you may discuss that in the comment box.